Hi everyone, thanks for coming. This is our second seminar series, so second seminar in the series. And I'd like to start off by acknowledging the traditional owners of the land and custodians of the land that we meet on today, the Wurundjeri people of the Kulin Nation. But I'd also like to acknowledge that our Indigenous community are one of the groups of individuals in our country which are highly afflicted by infectious diseases. And that's particularly relevant today to our talk about fungal infections and in immunocompromised host, where vulnerable populations, particularly Indigenous individuals, most highly prevalent recipients of cryptococcal disease and other fungal infections in Australia, and I think this is highly relevant to think about that today. So I'd like to introduce Monica, who is an infectious disease clinician. She works at the Peter Mac and the Royal Melbourne, and she'll be giving us a tour de force of epidemiology and immunology of fungal infections today. Thanks, Monica.
Thanks, Anna. Hello, everybody. I'm going to be talking about fungal infections, and these infections are obviously things that affect all the population, but more so people whose immune systems are compromised, particularly people who are getting chemotherapy, transplantation, or maybe getting monoclonal antibody therapy for various inflammatory conditions like rheumatoid arthritis, inflammatory bowel disease. But I'm going to focus mainly on the cancer transplant area, seeing that's the area that I work in. And I just want to open up by saying that fungal diseases have been a bit neglected, really, in the grand scheme of things. There's been publications over 10 years or more pointing this out. The first one was from one of the leaders at the CDC, talking about how these diseases are uncommon but are growing public health problems. And this was echoed again in another editorial in 2017, saying that, again, fungal infections are a bit ignored, but are really posing important biosecurity, food, public health issues. And I'll touch on that a little bit in the talk in relation to cancer and infection. But as you know, fungal infections encompass a wide variety of different pathogens, and this is one report looking at trying to actually get some kind of idea of the global burden of fungal infections and really focusing on systemic, opportunistic fungal infections. So the type of infections, I'm not talking about athlete's foot, for example, which is the most prevalent fungal infection worldwide, but really just causes inflammation and disease on the foot and nails. I'm really talking about systemic infections that affect the host's major organs, and opportunistic in terms of generally affecting people whose immune systems are damaged in some way or impaired in some way. So as you can see, the mortality rates and the prevalence of these infections, when you look at it as a global picture, is quite large. And over 2 million patients per year are estimated to die of fungal infections globally. And probably a large burden of those are made up from developing countries, and things like cryptococcal meningitis being very prevalent in countries where HIV is still a problem. So there's a very wide variety of different infections, and I'm going to focus today largely on aspergillus, which is the leading mould infection we see in people with impaired immunity. I'll talk a little bit about Candida as well, which is probably a second leading cause of infection in people with impaired immunity. And I won't really touch on cryptococcus. I may talk a little bit about mucormycosis as well. So when we look at the burden of fungal infections, this was some very nice work done in Australia by one of our PhD students, Karen Urbanchik, who looked at how much we're actually spending on treating fungal infections in major hospitals around Australia. And as you can see, quite a lot of money is being spent on antifungal drugs. In some hospitals, it's in the over $4 million a year budget range. So that's a large amount of hospital budgets being spent on treating infections. So what I'm going to talk about now is invasive fungal infections and some of the emerging issues around them. I'll talk a little bit about antifungal resistance, which is always a problem whenever we have drugs to treat an infection, the organisms always develop ways to become resistant, and this is no different with fungal infections. I'll talk a little bit about Candida auris because this has been a new emerging infection that's really only been noted over the last 9 years or so. And I'll talk about some diagnostic issues and some of the new things that are coming along to help us diagnose infections and treat infections better. So I'm going to start off with how I got into this area, which may be of some interest because you're all starting out your careers, and it's sometimes interesting to hear about how people ended up in the areas that they're working in. I was very interested in the immunocompromised hosts, so I decided to go to Seattle to work with Joel Myers, who was really a leader and visionary in saying that we have to develop specific skills and capabilities to treat the type of opportunistic infections that occur in people whose immune systems are being suppressed. And the Fred Hutch was really one of the places where bone marrow transplantation was first developed. And so obviously had quite a lot of experience with opportunistic infections such as CMV and fungal infections. So my goal was to go there and work on CMV, cytomegalovirus infection. And so I went off there to Seattle in the 90s, and as you can see, managed to have my first child from this very 90s picture in Seattle at the same time as working there. And what I went to do was work on CMV, and this is a picture of immunofluorescence for CMV inclusion bodies. What we used to call shell vial cultures before we had PCR. We used to have to culture the cytomegalovirus in tissue culture and then stain with fluorescent antibodies to detect it. And so I did some work on CMV, and this was really why I went there. But my mentor, Joel Myers, then asked me, do you want to be involved in a study looking at fungal infections? We have a study which we're developing to look at fluconazole, a new AZOLE antifungal drug, which has just been really developed, and we're going to look at it as a prophylaxis, a prevention against fungal infections in stem cell transplantation. So I had to think about it very carefully because I was really focused on CMV, but decided I would take up the offer to be involved in a randomised clinical trial. It sounded quite exciting. And so after that time, I really evolved into being someone who works on fungal infections, just due to that chance sort of opportunity and decision to take that up. So what we did was we looked at using fluconazole prophylaxis in people undergoing allogeneic stem cell transplant, and we gave the drug for 75 days, which covered the period of their neutropenia after a stem cell transplant and also the early episodes of graft versus host disease after their transplant. And we did see a marked reduction in systemic fungal infections and actually an improved mortality, which was very pleasing because not many things you can do in the setting of a highly immunocompromised patient who's undergoing a transplant can improve mortality. So what did we see in terms of infection? And this is very interesting and it's changed a lot. The leading cause of infection in the placebo arm were Candida infections. And you can see there was a couple of other mould infections, but not very much. Candida was the big problem. And this drug very nicely did prevent Candida infections. And we saw one other interesting observation, which really has only been explained in the last few years. We saw that, okay, people getting fluconazole had more grade 2 graft versus host disease, which is characterised by nausea and vomiting, and may actually have been not so much graft versus host disease, that's actual nausea and vomiting. But grade 3 and grade 4, the most serious types of graft versus host disease, were dramatically decreased in patients getting fluconazole prophylaxis. And that could never really be explained until recently. There's been increasing work on the microbiome, the gastrointestinal microbiome, and we are learning that Candida in the gastrointestinal tract, particularly overgrowth of Candida, can bind to receptors in the gastrointestinal tract and increase inflammation, thus making a nice milieu for graft versus host disease to actually develop and take hold. So interesting that a study that you do 20 years ago, you have to wait for 20 more years to work out the the mechanism of one of the observations that you see. But I guess that's the way it often is in science, basically. So what else did we see on this study? That we saw this survival advantage persisted for up to 8 years after the allogeneic stem cell transplant, which was a very interesting observation from an intervention that occurred early on after a stem cell transplant. And then subsequently, we looked later in the 1990s at the causes of fungal infection in patients undergoing allogeneic stem cell transplant, and things had completely changed. We were now using fluconazole prophylaxis, but what we saw now as our leading cause of infections were aspergillosis, occurring in about 8 or 9% of patients after an allogeneic stem cell transplant. So this sort of shows another principle, is that when you prevent one thing in a highly immunocompromised group of patients, there's always something else that can occur thereafter. And what we also saw in this study, we looked at various risk factors, and interestingly, ganciclovir was a risk factor. Now, ganciclovir is what we use to treat CMV, cytomegalovirus reactivations. So somehow there was this linkage between cytomegalovirus reactivations and fungal infections. Well, jumping forwards into the next sort of millennium, we did again look at fungal infections in our acute leukemia patients at the Royal Melbourne Hospital. And now we're using newer antifungal prophylaxis, not only to prophylax against Candida, but also to prophylax against aspergillus and some of the other mould infections. So things have changed again. Now what we're seeing is that our breakthrough infections are a variety of different organisms. We're seeing infections such as Lamentaspora, which is an unusual mould infection, and Candidas, again, resistant types of Candida, Candida glabrata. And we're still seeing aspergillus, but we are seeing this other pattern of infections as well. So again, important to recognise that the types of infections that you're going to see in these immunocompromised patients depend very much on what they've received, what kind of antifungals, what kind of medications they've received to prevent infections. But the mortality is still very high for patients with an invasive fungal infection, still much higher than those who don't have an invasive fungal infection. So still area that we really have to work on improving. Well, again, going back to this linkage between CMV and fungal infections, and again, it's hard to tease out because the patients, of course, who are the most immunosuppressed are going to be getting fungal infections, and they're also going to be getting viral reactivation like cytomegalovirus. But in this study, it did seem that CMV reactivation per se was a risk factor for going on to get an invasive fungal disease. Going back to that idea about virus potentially being immunomodulatory, also the antifungals that we use to treat this infection cause neutropenia and dropping the white cell count, so then fungal infection can become more of an issue. So I think that, you know, it's all interrelated. So it's not like a patient who are treating just one infection in isolation. Getting a viral reactivation then makes you more predisposed to get other opportunistic infections. And in this study, which was a very recent one in allogeneic stem cell transplant patients in Royal Melbourne and the Alfred Hospital, you can see that although Aspergillus fumigatus is still the most common mould infection, we're starting to see a very mixed bag of different moulds, and that moulds that you might come across very frequently causing infections. So things like Poecilomyces and other and Rhizopus, which is a mucormycete, an uncommon mould infection. So we're starting to see now in our patients quite a different profile of infection than what we might have seen 20 years ago. We're seeing a variety of different moulds. We're seeing Candida species that tend to be more resistant to antifungals. And just going back to that study about CMV and invasive fungal disease, this was the actual graph from the paper showing for patients who had a CMV reactivation, their rates of fungal infection were around 15% compared to those without CMV reactivation who had much lower rates of fungal infection. And these findings have really been borne out about using azole drugs to prevent infection and what types of infections you see in breakthrough by studies elsewhere. And this is just one example from Duke University Hospital, where they looked at the patients who'd got mould-active prophylaxis and those who hadn't got mould-active prophylaxis. And you can see here the mucorales, the mucormycetes, are much higher in those patients who got the mould-active agents. Other moulds are much higher proportion, even though overall aspergillus is still the leading cause of breakthrough infections. So we're seeing a very different pattern of fungal infections now due to our use of preventative medications. And I'll just talk a little bit now about some emerging issues in the area of antifungals. So this is some pictures from the Australian New Zealand Mycology Interest Group, which is a group of scientists and doctors and vets, actually, who are all interested in fungal infections and how we treat them. These are some pictures of my Westmed colleagues and David Ellis, who's the scientist who actually set up this group while he was working at the Mycology Reference Centre in Adelaide. But what you can see here is this is one of our biannual meetings, which we have usually in a very nice tropical environment in Australia somewhere. But during the 1990s and the 2000s, we did have a lot of new antifungal therapies coming on board. You can see, as I was talking about, fluconazole came on board around the late 80s, early 90s. Then we had itraconazole, an azole that also has activity against moulds. We had terbinafine, another different class of antifungals, which is the commonest drug used to treat athlete's foot, actually, because it's very concentrated in skin tissues. We had various different forms of amphotericin B. Now, amphotericin B has obviously been around for a long time, but these were lipid components of amphotericin B, and encapsulating the amphotericin B in lipids is a mechanism to try and reduce their toxicity to human mammalian cells. So these were an advance because they were less toxic drugs than what we'd had previously. And then we also had the echinocandin class of antifungal drugs, which is, again, a new class, which all came out around the 1990s and 2000s, giving us good options to treat Candida infections. So a lot of things happened over that time period where we had some new drugs to treat difficult infections. So these drugs have all been around now for 10 years or so. And what are we seeing in terms of resistance? Well, not a great deal so far in terms of Candida resistance, although the pattern, again, I've talked a lot about the pattern of mould infections, the pattern of Candida infections has also definitely changed in that we're seeing less Candida albicans, which is the very common form of Candida, not so difficult to treat, and more of Candida glabrata, which is a more resistant Candida species, often resistant to the azole antifungals, which are our first-line drugs. So there has been a slight change in these national studies, surveillance of fungal infection and Candida. We can see this over this sort of 10-year period, that the types of Candida species we're identifying, and these are all from blood cultures from patients with Candida, are changing, and changing towards a more resistant, intrinsically resistant type of Candida. But when we looked at the antifungal susceptibility in these studies, we didn't really see a huge issue with the echinocandins. And as you remember, they're the drugs that came out in the 2000s. So we are now starting to see more echinocandin-resistant Candida, but it's still not a large problem. But in some parts of the US, in some hospitals where they widely use echinocandins, 10% of their isolates are echinocandin resistant. So it's something that we really have to look for in the future. As for azole resistance, there was a survey done from the Mycology Reference Laboratory, taking out all the aspergillus samples that had been sent to them and screening them for resistance against azoles. And as you can see, only 2 were found with a marker for resistance. And this is the marker that I'll talk a little bit more about in a moment. This mutation in the CYP51A enzyme is really much a marker for environmental azole exposure. And this is the type of azole resistance which is occurring and particularly been highlighted in Europe, where in Holland now about 15% of clinical isolates have this marker and are resistant to the azoles. And this is a big deal because the azoles are the first-line drugs we use to treat aspergillus infection. We use voriconazole. And these types of mutations impart voriconazole resistance. So I'm going to talk a little bit more about antifungal resistance, but I'll just put a picture in here. I'm not a botanist, but there's been quite a lot of publication about the largest living thing on Earth being a fungus. And there's this area in Oregon where fungus is just growing throughout the soil, throughout the trees, everywhere, and it's thought to be one organism. It's just visible in different parts. It's visible in the ground, it's visible in the trees, but it's all one area of fungus. So just again, I think showing the importance of fungus in our ecology. But what about this azole-resistant aspergillus that I've talked about? It has been identified primarily initially from Europe, but subsequently from many other countries internationally. And this, because azoles are the first-line treatment, if you don't recognise that you've got an azole-resistant aspergillus and you just treat the standard way, the mortality is around 88%. So just about all those patients would die because it wasn't recognised that they had a resistant infection. But what's interesting about it, we can't predict who's going to have this infection because if you're in an area where it's prevalent in the environment, you probably acquire it from the environment. So it's seen in people who've never had antifungal drugs previously, but have acquired it from breathing in the spores from the aspergillus in the soil or in the air around them. And as I mentioned, now it's about 15% of clinical isolates in the Netherlands. And there's been very nice studies done to actually genetically link the type of invasive infection to the environment source of infection. And there's been some work done also looking at how you create this fungus, and you can create it by putting the azoles into the soil. And azoles are widely used in agriculture, actually, and very widely used in the Netherlands in the tulip growing industry. And it's thought that this close proximity of people and agricultural use of azole antifungals is what's fostering the resistance, and why it's particularly seen in the Netherlands, not so much seen in Australia and less seen in the US. Maybe people have postulated that it's the intensity of use in a very kind of closed space or close proximity to people, whereas our agricultural use might be more remote from the population centres. But interestingly enough, it has now been identified in animals, the azole-resistant aspergillus. So this is a scan of a dolphin. There is a case report now of azole-resistant aspergillosis affecting the lungs of a dolphin as well. So it's spreading from the environment to humans, but also to animals, which is a great concern. So this is a kind of publication that tried to explain this whole evolution of resistance in the azoles and aspergillus about killer tulips. And it's not really the tulips, it's the antifungals that are used to protect the tulips as they grow. So I was going to talk a little bit about Candida auris, because that's another topical outbreak that's occurred globally and really has now affected us also here in Melbourne and in Australia. And I've talked about Candida, but Candida auris is a new species that's probably only been identified as a cause of disease in the last 9 years or so. Prior to that, it was really unknown as a cause of disease. So something's actually happened to make this Candida emerge suddenly as a global problem. And it's really a fascinating exercise to figure out why this might have happened. I'm just going to talk a little bit about fungal outbreaks as well, and we'll get on to Candida auris in a moment. But in terms of fungal outbreaks, if you work in a hospital or an area that looks after patients, you're always concerned about building works, about demolition, because often in the dust and in the ceilings and in areas like bathrooms where you've got damp areas, mould can grow very easily. And so as you knock down these old buildings, mould can be released into the environment, the spores, and patients, of course, will inhale them. So there's been a lot of work done in our hospitals and with protocols now to strongly protect patients from any of the dusts that are generated when you knock down buildings and hospitals. And there's a lot of work being done currently because of the digging for the new metro tunnel to prevent those potential dust containing spores then being inhaled by patients. So as you can see, there has been a decline in outbreaks around immunocompromised patients due to fungal spores and fungus during construction, probably, hopefully reflecting some of the good work that's been done, developing protocols and mechanisms to prevent patients from being exposed to the dust. But there are a number of new outbreaks that have occurred, and if we look at some of the mould outbreaks that have occurred in hospitals, these are very interesting because they're often related to environmental contamination of things that we might not necessarily think about. So optical lens implants of PD, peritoneal dialysis fluids, medical equipment, but also we're starting to see mould outbreaks occurring as a consequence of natural disasters like tsunamis and earthquakes, where water and soil are contaminated and then with fungus, and then contaminate wounds of people who are injured in these natural disasters. So change a little bit in the types of outbreaks that we're seeing, and often these types of outbreaks do involve mould infections other than aspergillus. But there was one recent outbreak which is quite interesting in some of the transplant hospitals around Pittsburgh, and where they tracked this outbreak down to eventually, and it took quite a while to unravel where the mould was coming from that these patients were developing after undergoing heart-lung transplants or liver transplants, and then developing serious opportunistic mould infections. And what they found was that there was a common linen processing plant, and this processing plant was quite old. The roofs were very dusty. Steam was being emitted due to all the washing and cleaning of the linen. And then this was kind of settling and picking up bits of the dust and settling back down into gaps in the roof, into the building where the linens were being stored. And so they thought that the linens were actually the cause that were being then transported to the hospital and used for the patient's beds, etc. And this prompted another study to look at linens in the US hospitals, and they looked at 15 different sites. And you could see that at 20% of those that they tested, they did find mucorales or mucor, and they also found aspergillus and other fungi. So this is an interesting sort of concept that now there are kind of other sources for the mould and for mould outbreaks that we really need to be able to investigate. It's not all just related to knocking down an old building, for example. So I was going to talk about Candida auris, and this has got a lot of attention because it's quite drug resistant. It's often resistant to all the azoles. It's sometimes resistant to amphotericin B and sometimes resistant to echinocandins. So you can see that this drug could potentially be resistant to all of the antifungals that we've got to treat. So it's interesting because it behaves more like a bacterial than a fungus. We're usually with Candida, we usually know that Candida usually colonises the patient, lives in the patient's gut, in the mouth, on the skin. But this bug can be on the skin and highly shed from the skin and then picked up by other patients who come into the patient area that this person has been in. And it can also live on equipment, like one outbreak was linked to auxiliary temperature thermometers that were being used on patients. So it can spread very quickly within a hospital setting. And of course, again, most of the people in the hospital setting are immune compromised in some way and quite ready to kind of be a host for this type of infection. It hasn't really been found to be spread amongst hospital staff who are generally healthy, but more so between patients. So this was what I was talking about earlier, very interesting publication in 2017 looking at how this emerged simultaneously on 3 continents, Africa, Asia and in the US as well. So this was very interesting as to how this actually came about. And when people have gone back through culture collections, it's really not been found in very old isolates of Candida in collections going back 20, 30, 40 years. So it seems to be something that's really emerged in the last 10 years or so. And the question is why? It is a highly drug-resistant Candida, so perhaps it's been facilitated by a use of antifungal drugs, but there may be other explanations that we're just not aware of. So it did occur here in Australia. The CDC keeps a map and updates the map regularly of where it's been isolated and where transmission has occurred around the world. And now Australia has got itself a red marking to show that transmission has also occurred in Australia. The first case actually occurred quite a few years ago in Perth, but just wasn't publicised at all, and has only been recently written up and reported. And then there were several cases in Melbourne last year and a couple of cases in Sydney. But what's been common largely amongst the few cases we've had is that they've generally been imported. People have come from healthcare facilities overseas and possibly brought this bug with them. But of course, we had a case of it, 2 patients who were colonised with it in Melbourne, and that created a lot of new kind of probably fear in the public about this superbug now being in our Melbourne hospitals. But just to talk about maybe the 2 patients that were detected with it, some common features of these patients were that they had very advanced malignancies. They'd gone through a lot of chemotherapy treatment. Both of them had received care overseas in hospitals, one in London, one in Dubai. And, of course, had very stormy courses with multiple hospital admissions and had also received fluconazole prophylaxis at some point in their care to prevent fungal infection. And then they were identified to have Candida auris, which was a colonising organism and didn't cause an invasive infection, but was still a large concern to the public health authorities, who actually wanted us to screen all the patients who'd been in contact with these 2 patients. So over 70 patients were screened and none were found to have this organism. But I think it just shows that it is now in Australia and we have to be aware of it. This, again, was susceptible to the echinocandins, so we did have a treatment if we had needed to treat it. And the MDU did some genetics looking at the 2 cases that we had. And as you can see, there are these distinct kind of clades of infection varied by different geographic areas, so South American, Japan, South Africa, India, Pakistan, which are fairly distinctive. So the MDU were able to find out that these two patients, probably one transmitted it to the other, because there was only 3 SNPs different between the two isolates, meaning that these were very closely genetically related. We don't know how the transmission happened, we don't know which one transmitted it, but this is a very interesting finding, I think, and that they belong to the India-Pakistan clade, which is probably the commonest one seen in some parts of the US, and in Europe and in Arab regions. So it's a bit hard to say which patient might have brought it back. So I think outbreaks really tell us we need a strong public health approach. We need to be looking out, we need to be capturing cases of fungal infections so we can identify when we've got an outbreak. Rapid testing is very helpful so that we can treat patients appropriately very quickly. We've got to be able to identify it quickly as well and recognise who's at risk. So now the Department of Health will be introducing a policy or a suggestion that all patients transferred from overseas healthcare facilities are screened for this pathogen. Well, briefly talk about diagnosis, and I think, we have to go back to basics, and one of the key drivers of antimicrobial stewardship is being able to make a rapid diagnosis. If we don't know what we're treating, we're using a lot of drugs indiscriminately. And so fungal infections are no different. Where you don't know what you're treating, you've got a patient who you think's high at risk for a fungal infection, you haven't identified a pathogen yet, you may be using broad spectrum antifungal drugs for some time before you actually come up with the correct diagnosis. So one of the things that I think is really critical is that we're able to diagnose these infections much more rapidly. And this has also got an impact on patient care. And this is only one study looking at time to treatment of Candida in the blood, CANDIDEMIA. And if you delay treatment beyond about 12 hours from the time that this infection starts, you're going to see a higher mortality in patients. So there is a real driver, not only for stewardship, but for patient care to be able to treat and identify these infections as quickly as possible. And a number of authorities have talked about how we need to improve our diagnostic capability. And this is from a paper talking about stewardship and saying that at a major hospital, we should be able to do these kind of diagnostic tests. But just to let you know, not all major hospitals in Australia can do these diagnostic tests. So we're still not there in terms of having the kind of diagnostic facilities that we need to rapidly identify these infections. Well, how do we identify these mould infections in clinical practice? Candida's not so hard. We can identify it from the bloodstream, but it does take 5 to 7 days to grow on traditional cultures, sometimes 24, 48 hours, but more likely 5 days. So again, rapid diagnostic tests are very important for Candida. And for mould infections, as they usually are breathed in and land in the sinuses or in the lungs, the spores, and then cause the infection, we generally look at the respiratory tract for our diagnosis. So we generally try and get a biopsy, or if we can't do that, getting a lavage, sending some fluid down a tube, bronchoscopy that's put into the lungs, and sucking it back out, and then looking for markers of fungal infection in that fluid. And although these tests are good and help us diagnose infection, again, we're a bit lagging in being able to access them at all hospitals. This was a study done through the ANSMIG group, and you can see that for these new rapid diagnostic tests like antigen testing or PCR testing, that most hospitals wouldn't get them back for 3 to 5 days or maybe even longer. So again, we're kind of a bit delayed and a bit behind in accessing these new rapid diagnostic tests. And it's actually the same in the US. This was a survey of major transplant centres in the US, just to focus in on some of the fungal molecular tests. Mainly they were send-away tests for the majority of centres, you can see. And these are transplant centres. So although the tests are out there, they're not that accessible to the clinicians treating the patient. The galactomannan and PCR are now both kind of ensconced in standardised definitions, and there were two large studies, one in Australia and one in Spain, looking at incorporating an aspergillus PCR into the way we treat patients and showing that we could diagnose infections earlier. We could sparingly use antifungals. If the tests were negative, we could withhold the drugs, and it didn't make any difference overall to patient mortality. So we know that the tests can be used effectively. It's just a matter of implementing them now into practice. And one of the big things to help will be these new definitions, and these standardised definitions of fungal infection. You might say, how's that going to help clinicians having a standardised definition? But now Aspergillus PCR for the first time has been incorporated as one of the standardised ways of defining what a fungal infection is. So it's very likely now that we will see these tests being taken up more widely now that they're kind of got the stamp of approval as being part of the way we diagnose an infection. Well, I've kind of spoken a bit about the non-culture-based tests, and the reason that they're so important is that cultures don't perform very well. I told you for Candida they take quite a while to turn positive, even in a blood infection, but even in bronchoscopies where you're actually sucking the fluid back out of the infected lung, only about 50% of the time do you get a positive culture on that. You might see hyphae under microscopy, you might see the antigen marker, you might detect it on PCR, but only about 50% of the time will you get a positive culture. I'll skip over that. So the last few slides, I just wanted to talk about what's new in the area and what are some of the emerging problems. And I think one of the big new areas, especially in the cancer therapy, is targeted drug therapy. And of course, there are a lot of new protein kinases being developed for treating cancer patients. And one of the areas that's really big is ibrutinib, which is a BTK inhibitor. But interestingly, after marketing, this drug had been associated with increased risk of fungal infection. And the mechanism's still being worked out. But of course, there is some early work with mouse models saying that fungal infections are more severe. There are also some mechanistic work underway to look at what ibrutinib does besides being a BTK inhibitor, how it affects other arms of the immune system. So there's a whole interesting area with the new drugs that are being developed, perhaps creating new niches for fungus. And what was important in the types of infections that these were seen in these patients receiving ibrutinib was that they were disseminated, they were aggressive, they were occurring in different sites like the brain compared to what we commonly see in the lung, and disseminated cryptococcus was occurring. So quite a different spectrum of infections. And so this is really an area to watch, I think, these new agents and how they impact. Ben Tay at our centre did a retrospective review of patients treated with BTK inhibitors, and you can see that the rate of fungal infections was about 12%. That may not be truly reflective because obviously when a new drug comes out, it's given to the most severe people who've gone through all the other options and have no other options. They're the ones who get the drug first. So we may be seeing a biased population selection here, but certainly we saw a lot of infections and quite unusual infections such as Lamentaspora as well. And one other group of patients we need to think about are those receiving checkpoint inhibitors, immune checkpoint inhibitors. Very common, used in multiple solid tumour groups now and also in liquid tumours as well. And so this is just an illustrative case of a 68-year-old man with metastatic melanoma who was receiving checkpoint inhibitor therapy and progressed and was changed from pembrolizumab to ipilimumab and developed the immune-related adverse effect of colitis, which is a commonly seen immune adverse effect from the checkpoint inhibitors, so got treated with high-dose methylprednisolone. And several weeks later, he complained of a cough and underwent X-ray, and you can see that he has a cavity in his lung there, and it was seen again on CT scan, and a biopsy showed that it was full of mucormycosis, a fungal infection. So melanoma patient, not someone we'd usually think about fungal infections in, but receiving a lot of additional immunosuppression due to the checkpoint inhibitor therapy. Other new things on the block are CAR T cells, again a new advance for treating lymphomas. And of course, one of the common side effects is the cytokine storm that these T cells can induce. And so these patients will also be receiving high-dose prednisolone and tocilizumab, which is being found to be effective in dampening down the cytokine storm. So again, another group of patients who we may be starting to see fungal infections in. And yet a third group are those in the ICU with influenza. And the Dutch have been very proactive and done a large survey of patients admitted to ICU with influenza, showing that up to 20% of them will develop an invasive aspergillus infection of those patients who need to go to ICU and be ventilated because of pneumonia. So again, these other groups of patients that we're seeing. I won't spend too long on the last couple of slides, but just to show you that there are some new tests in train and are being evaluated. Most interesting is a direct PCR test to detect this resistant gene to azoles in aspergillus, which you can use on bronchoalveolar lavage. And of course, new ways of testing antigens, like using a single-use test like this, like a pregnancy test, to detect the aspergillus antigens in blood or bronchoalveolar lavage fluid. And we do have some new classes of drugs, and we will be having these. This is already in study, this erotamide class of new antifungal agents, and there's a new type of echinocandin-based agent as well that can be given once weekly. And this will be in study very soon, and this is currently in study at our hospital. So we do have some new classes of antifungals coming, which may address some of the issues that we have with these highly resistant pathogens. And lastly, we do have an NHMRC grant led by Kaz Thursky at Peter Mac and Royal Melbourne to look at antifungal stewardship, because we've talked a lot about how whenever you use antifungals and if you're using them indiscriminately, you're likely to develop resistance. So this grant will, over 4 years, look at the processes that we use to identify use of antifungals as appropriate or inappropriate. It'll look at how we're going to develop some structured surveillance systems, and it will look at outcomes of use of antifungals. So that's a kind of exciting area for the future. So just in closing, I guess I'll summarise by saying that fungal infections, they're still occurring even though we give patients preventative antifungal medications. We're seeing both Candida and Aspergillus, the commonest ones, becoming more resistant to antifungals. We're seeing new types of outbreaks. Candida auris is a typical example of a new fungal species that's appeared, but also non-Aspergillus molds like mucormycetes. And we really are still a bit limited in how we diagnose these fungal infections. There are a lot of new immunosuppressive therapies that may be creating new opportunities for fungal infection being used in our patients. And really, we've really got to start developing systematised surveillance and antifungal stewardship, as well as good public health responses. I'll finish there, but just to say in Melbourne in 2020, we'll be hosting the Infections in the Immunocompromised Host meeting, which will be on in Melbourne. So please, if you're interested, please come to that. And just to say, if you want to come and talk further, we're in Peter Mac, and this is the team. Thank you.
Thank you, Monica. Any questions?
You mentioned the high mortality rates in azole-resistant patients. How difficult is it to include diagnostic tools that indicate resistance?
Yeah. So if you're in Europe, like working in Belgium or the Netherlands, that's already being included. So those probes for the diagnosis of the azole resistant, because it's usually a few limited mutations, they've been included now in the diagnostic PCR tests that are applied to the bronchoalveolar lavage fluid. So that's good. In Australia, because it's not a widespread problem at this point, most labs won't include it because of cost. But, you know, if it were to become more prevalent, there is an option that we could include those tests.
What do you think is the cause of the rise of these new species coming out? Is it because we're improving our diagnostics and thus they're already there and we can see them before?
That's true.
Or is there a change in diet which is influencing microbiome, thus influencing fungal colonization?
Yes, I think it's a great question. We really don't know, and there may be multiple things at play. I think the microbiome is a really important area for future work, and we really haven't got onto the fungal microbiome yet. We're still looking at the bacterial microbiome. So I think we don't know if what's happening in our bodies, in the areas that were colonised with fungus, what changes are taking place. But certainly, in a global sense, use of antifungals certainly drives resistance as well.
And so you have this dolphin, right, that has lung disease. How and why, once it gets infected with a strain of a mutation, does it develop disease? Do we see this kind of disease where a Parkinson's mutation is some kind of condition in the
Yeah, I think animals do get aspergillus infection and do get fungal infections. A common one in Australia is the koalas with cryptococcus, because they have very large nasal cavities and are inhaling the spores from around gum trees, etc., and do develop the infection. So I think, again, dolphins were affected by the cryptococcus outbreak in Vancouver Island. So I think, yes, animals definitely get fungal infections. Maybe we just don't look hard enough, you know, to see how prevalent it is and whether they are being infected with resistant organisms. Unless they get to a vet, they're probably not tested.
I may have missed it, but is there a reason why we don't implement like Eliza tests on products as like a screening test?
Yeah, so you're looking for the fungal antigens in blood products or, I think generally the cost probably. I mean, you could do that, but I think we really haven't seen outbreaks related to blood products. Where there have been outbreaks related to fluids, it's usually been that they've been contaminated during some sort of natural disaster where their normal kind of sterile storage and the chain of storage has been disrupted in some way. So I don't think it's very common to see these infections due to contaminated products unless you have some catastrophic event usually. And then, yes, you could apply these tests to fluids. In fact, it has been done, and that's how some of the determinations that you sometimes get in a patient's serum false positive results due to the plasma that they've received or the resuscitation fluids. So you can find very small traces of fungal antigens in various things like antibiotics, like plasmolite and things like that, yeah. But they don't usually cause the outbreaks or the infections. It's usually where you've got grossly contaminated infusates. Thank you.